Hello, I'm your instructor, Leslie Sayre, and here's a quick and dirty review of Unit 4, Cell Division, Chapter 16 if you're following along with the textbook. I'm going to pop up some slides showing images for this unit, which is probably more useful than looking at me. Okay, starting with the cell cycle, you can see from this graphic that much of the life of the cell is in gap 1 of interphase. In fact, a cell will spend its entire life in this phase going through its normal routine. You know, if it's an endocrine cell, it's going to be making hormones. If it's a nerve cell, it's going to be conducting action potentials and so on. It's only if the cell needs to divide that it is going to move from gap 1 into S phase or synthesis phase of the cell cycle, and this is when the DNA is replicated. So if the cell is to divide, whether it's by mitosis or meiosis, the DNA needs replicating, doubling up. Now if it didn't replicate, when prophase comes around and the chromatin condenses, it won't form nice sister chromatids. It'll just be a single chromatid. And how does a single chromatid get shared between two cells after dividing? So whether mitosis or meiosis, the DNA is replicated once. Now I know meiosis has 2 divisions, but trust me, replication only occurs once. Now when that's done, the cell goes into gap 2 to bulk itself up for when division occurs. Now this graphic is showing cell and cytoplasmic division. Basically, mitosis involves organizing a pile of DNA from one nucleus and into 2 piles of DNA and then forming a nucleus around each pile. That's mitosis. Cytoplasmic division occurs right after, and that's when the cell itself physically splits into 2, each with its own pile of DNA. Now you need to be able to identify which stage of mitosis you're looking at by way of a diagram or a photograph. Now your textbook has both, so I suggest you give it a look-see. The 4 mitotic events are prophase, metaphase, anaphase, and telophase. And in prophase, you'll see the chromatids have condensed, and what's more, they're stuck in the they're stuck to their identical copy at a position called the centromere. Now in prophase 1 of meiosis, you'll see synapsis, and this is the coming together of homologous chromosomes. So this doesn't happen in mitosis, but in meiosis, homologous pairs, they get together. Each chromatid begins wrapping itself around its non-identical partner, called crossing over, forming a structure called a tetrad. Now the purpose of this is to create recombinant chromatids. Recombinant just means different from parents. So where on the chromatid the crossing over occurs is going to be an exchange of DNA between the homologous pairs. So instead of a chromatid having only alleles from one parent, and at the end of prophase 1 it's going to have some of the other parent's alleles on it too. Now this means each chromatid will end up with a unique combination of alleles. And should that chromatid be a part of, a sex cell, for example, that gets fertilized, then the zygote will carry a unique genetic profile, which is important for the survivability of the species, enabling them to adapt to changing environments. Now the longest stage of mitosis is metaphase, and that's where the chromosomes align along the equatorial plate of the cell. And if a karyotype is to be performed, to analyze the chromosomes, it will be during this phase. Now, studying the DNA of any organism requires we look at the diploid cells with homologous pairs of chromosomes. This is a karyotype of a normal female human. See the 2 X chromosomes? So the chromosomes are arranged beside their homologous partners. The numbered chromosomes are called autosomes. The X and the Y, which is not shown because this is a female, are the sex chromosomes. So there's a medical procedure called an amniocentesis that's going to be performed on pregnant women, so they can take a karyotype to assess the chromosomal health of the fetus. Metaphase 1 of meiosis sees another attempt at creating recombinant products. The chromosomes are still in the form of tetrads. So as you can see from this diagram, the green and the blue chromosomes are orientated on the top, and the yellow and the red are on the bottom. So the blue and the green are going to separate together to their own cell, and the red and the yellow are going to separate together to their own cell. So two cells after the first meiotic division, one with blue and green chromosome combination, and then one with yellow and red chromosome combination. But this is arbitrary. There is a equal probability that the red and the blue could end up in a cell together, and the yellow and the green could end up in the cell together. So this is independent assortment. This is another valuable form of variation. Now you might be, you might think that, well, with two homologous pairs like this, what variation? But humans have 23 homologous pairs, and that makes over 8 million possible combinations. Okay, this is important. Most of you know that there are two divisions that occur in meiosis, resulting in 4 haploid cells, the sex cells. The first, meiosis 1, sees the separation of homologous pairs. Now, up to this point, the homologous chromosomes are still arranged as tetrads, remember? So when we get to Anaphase 1, the spindle fibers are going to separate those homologous pairs, pulling them toward the poles of the cell. Then, following cytokinesis, both daughter cells divide again, meiosis 2. The chromosomes in these cells do not have homologous partners. Chromosome number 1, for example, is in one daughter cell, and the homologous chromosome number 1 is in the other daughter cell. One X chromosome is going to be in one cell, the other X chromosome is going to be in the other daughter cell. You get the idea. So these are then haploid cells. They're chromosomes without their homologous partners. But the chromosomes are still arranged as sister chromatids. So the second meiotic division, just like anaphase in mitosis, sees the sister chromatids separate. Now, nondisjunction is a type of abnormal meiosis resulting in gametes with an incorrect number of chromosomes. So if any one of those 23 homologous pairs don't separate at anaphase, then the resulting gametes are all impacted. 2 of the 4 gametes are going to end up with a missing chromosome because it's now found in the other 2 cells. Not a big deal unless one of these cells is fertilized. So if one of these cells gets fertilized by a normal gamete, the resulting zygote will either have single chromosome number 21, say, instead of a homologous pair. That's called monosomy 21. Or we'll have an extra chromosome 21 in addition to a homologous pair, as in trisomy 21. It's important to clarify the reason for reduction division. See, the first meiotic division, going from diploid to haploid, half of the chromosomes in your somatic cells you inherited from your dad, the other half from your mom. So the process of making gametes reduces chromosomes from having homologous pairs to ending up with single chromosomes. So where there was 23 pairs in humans, there's now just 23. And that's because the gamete will fertilize another person's gamete, combining the chromosomes in each nucleus, creating a new combination of homologous pairs, which is the third mechanism of variation. See, there's crossing over, independent assortment, both happening in meiosis, and fertilization. So know how meiosis and mitosis are similar and how they're different, and don't fall into that trap of diploid cells divide by mitosis and haploid cells divide by meiosis. None of that is correct. Primary spermatocytes, they're diploid, and they definitely divide by meiosis. And think about it, do sex cells even divide? No mammalian sex cells that I know of. So it's better to say this: diploid cells are products of mitosis. Haploid cells are products of meiosis. Okay, following fertilization, the zygote undergoes several rounds of mitotic cell division before implanting. Now, if all of the cells prior to implantation are identical, and should a group of these cells split away and implant separately, then you're going to get identical twins. Now, you'll certainly see many questions on life cycles, particularly the alternation of generations life cycle. Diagrams are going to vary, but basically a diploid organism will produce gametes by meiosis, and a monoploid organism will produce gametes by mitosis. All plant life cycles include a sporophyte generation and a gametophyte generation, but one generation is typically dominant over the other. For example, vascular plants like the fern shown here are predominantly diploid sporophytes, while non-vascular plants like mosses, for example, are predominantly monoploid gametophytes. And in case you've not come across the word monoploid before, haploid and monoploid involve cells that don't have homologous pairings of chromosomes. So you call these cells haploid, but the organism that make up these cells is monoploid. Okay, well, I hope that's helped. I mean, as usual, please email me any questions that you have, and if you like, I'm very much open to offering tutorials for anybody who wants them. So, good luck with your exam.